All right. Today we're going to be doing a so-called masterclass on maternally loaded RNA. So a lot of students in the lab have been asking, or have been asked, about why we're doing 2 crosses in order to get RNAi in the early embryo. And so what that looks like is virgin female with the RNA that you want, cross to male with the Gal4 line that you want, and then using the females that are double hets and back crossing them. Let me back crossing to the RNA line. So males that are homozygous for the RNA that you want. Now, the reason that you do this is because at this stage, there's no way to get maternally loaded RNA from either direction that you do this, with the males with the Gal4 or with the females with the RNA under the UAS promoter, right? Because this is Gal4 driven. So the reason for that is because, let's just consider for a second, like if we do it this cross, right? Obviously the RNA is not going to be maternally loaded because it's not being expressed anywhere, right? It's not being transcribed anywhere because there's no Gal4 in the female, right? So in the maternal loading, there is no Gal4 in order to express the RNA. And if you do it the other way, then the maternal cells, the maternal diploid cells, have no RNA to express in the first place, so obviously it won't be maternally loaded. Now, then, when you get here, now the females have both RNA, the UAS RNA, and the whatever maternal driver Gal4 in all of their cells, right? In all of the diploid cells. So the diploid cells that are preparing the haploid embryo are going to have both. And since we know that this maternally driven Gal4 is expressed and maternally loaded, then we also know that it's going to be activating expression of the RNA that you want. So the RNA is also going to be maternally loaded. That also makes the Gal4 a bit maternally loaded and a little bit earlier acting if you're wanting to do some transitionary studies, during the maternal zygotic transition. So there is some Gal4 that's maternally loaded to activate the genomic RNA, the zygotic RNA that you're wanting. So that's the reason for that. And a test of this system.